The next speaker will be Martin Borch Jensen, who will give a talk by Zoom. Welcome, Martin.
Hi, Lena. Lena is my PhD advisor from some years ago.
Yeah, welcome.
Cool.
Go ahead.
So, yeah, thank you, Lena. Thank you, Morgan and Alex, for inviting me to speak at this great conference. I'm here representing Gordian Biotechnology, a company that I started almost 4 years ago, and which is in San Francisco, where I'm calling in from. Unfortunately, I couldn't make it in person. But I'm not going to pitch the company. I'm going to talk about how we make the research that we do most relevant for human aging, right? So how do we lead it towards eventual translation towards something useful, as Matthias was already doing, of course. So Gordian is a company founded on the somewhat simple idea that a lot of what happens in aging, whatever aging means to you, happens inside of a living organism and is not really well represented in cell culture. So interactions between different organs, different cell types, stiffness, circadian rhythms, all of these things that happen inside of an organism and that change with age. And so in order to do drug discovery that fully captures the complexity of aging, Gordian does screening for targets that might affect different age-related diseases, and we do that screening in high throughput and in living organisms at the same time. The way we do that, just as a brief overview, is that we generate barcoded libraries of gene therapies with payloads that either up or down regulate different genetic targets that we think might be therapeutic. And in these gene therapies, we have DNA barcodes that uniquely identify each individual therapy. We then take this library and inject it into an animal at a low dose so that whatever tissue we're studying becomes sort of a chimera with a few cells having received different interventions, but most of the cells in the organ having received no intervention and therefore still representing sort of the dysfunctional, old, sick, disease environment where we want our therapies to be functional. We then use single cell sequencing to pull out all of those cells that did receive an intervention, basically ask them two questions using the full transcriptome as a phenotypic readout. Question one is, which intervention did you receive based on sequencing the barcodes? And then question two is, along many biological axes, do you feel more like a healthy cell rather than the old and sick cell that you started out as? So this is the core technology we have. I'll return to that later in the talk, but I won't go into a bunch of details about this in the presentation here. Instead, I will, despite me owning a t-shirt that says In Vivo Veritas, I'll talk about how simply going to an in vivo system does not really guarantee that your therapeutic discovery in our case, but also in basic biology studies, are going to be relevant for human aging and human disease. And I'm going to use osteoarthritis as one of the diseases that we're working on as the example throughout, but the same holds for other areas of biology. So osteoarthritis, most frequently happens in the knee joint, is a disease with a lot going on. You have some combination of genetic environmental risk factors as well as load on the joint, leading to a degeneration of the cartilage that sort of lubricates the two bone structures, and death of the chondrocytes, which are the cells embedded in this extracellular matrix and that are responsible for making this cartilage. These cells are dying. Other cells are picking up signals of death, are getting inflamed in the joint capsule, and then immune cells are infiltrating. So you have this sort of multi-cell type interaction at work, which leads to progressive disease that happens over years and just gets worse and worse with age. So how do you model all of this stuff? What are the important factors? And so I'm just going to run through a few, and then I'm going to talk about how we think about that at Gordian and sort of offer some tips and even some helpful resources. Starting off, osteoarthritis in humans is very much age-related. You can see the graph starts at 40 here. There are a few cases of osteoarthritis in people younger than this, especially after sports injuries, but its incidence goes up with age. It's the #1 risk factor. But even on going in vivo, the standard model for studying osteoarthritis is to take mice, often young mice. This is one of the good papers, so sometimes older mice, and then perform a surgical intervention by damaging one of two tendons, either the anterior cruciate ligament or the meniscal median, and so destabilizing the knee, which leads to pressure in places, inordinate pressure in places that isn't usually there, and then you get this degradation of the cartilage. But even though you end up in the same place with this destroyed cartilage, are you getting there in the same way in this case, right? If you don't have the aging aspect, and if it happens very rapidly over a couple of weeks rather than over several years. Another question would be, since we know that immune cells infiltrate and sort of drive part of this disease, do you want to have a human-like immune system? And if you're using mice, as probably many people here know, the immune cell composition, in mice is very different than in humans. It's largely lymphocytes, whereas in humans you have mostly granulocytes, neutrophils, making up most of the circulating immune cells. Does this matter? I don't even know the answer, and I'm not going to try to give answers here, but I'm pointing out that even though we're in vivo and even though this is sort of translational, we might have species-unique artifacts, which of course is, people are no strangers in drug development to mouse-specific artifacts. Moreover, we're looking at individual cells, chondrocytes, let's say, but what about the organ? What about the structure of the organ? And sometimes just size matters. In the case of cartilage, you can see some stains here of where the cartilage, as opposed to the bone, is stained in different species. Mice are not even on here, they would be down here in terms of thickness. You can see that horses have a thickness that's similar to human, and they have layers of cartilage that's similar to humans. You can go through the same exercise for lots of other organs, like the heart or the lungs, where there's anatomical stuff going on. And if you are much smaller, then just the pressures in absolute numbers that cells are subjected to are much different. You could also ask, in the horse and the human, is the composition of the cartilage similar? Which may be important if you're studying, how are these chondrocytes getting signals from the extracellular matrix. And then finally, of course, cellular behavior do matter, cells do matter, even though non-cells also matter. There's lots of different ways you could characterize whether the cellular behavior in disease is similar in your model. We do a lot of single-cell sequencing, so I'm showing some single-cell sequencing data here, but it's really just representative of thinking, okay, we can look at the histology slides and see whether, the same gross changes happen in our model as in the disease, but are they driven, as we were talking about earlier, are they driven by behavior in the cells that is similar? One way you could do this is just plot out all the different substates of chondrocytes that are present in the joint, in osteoarthritic joints versus non-osteoarthritic joints, and see whether you can detect similar cellular programs with changes in gene expression across the severity of the disease. And so, as the example for osteoarthritis, just talking through what is Gordian doing and why are we doing that? What we're doing is going out and finding horses that have spontaneously developed osteoarthritis over a number of years, and then running our screens in that environment. And the thought process that we have here is that we know that there is a strong immune component, both resident macrophages and circulating immune cells, that is important to driving this disease. And so we wanted an in vivo model rather than an in vitro model in order to study the disease. If that's not present, if immune cells are changing chondrocyte behavior in a way that drives disease, and we leave the immune cells out, then how do we know that the therapies we're finding aren't simply, they would have worked in healthy chondrocytes, but in the actual disease environment, they do nothing. We also wanted the disease to take place over a number of years. However, we did not want to wait a number of years, and so that's why going out and finding spontaneous disease models that are already alive made a lot of sense to us. And part of the reason why we wanted this to happen over years is, again, going back to the incidence of disease being age-related, and I think this is relevant for pretty much any of the age-related diseases that are the biggest medical burdens, which is acknowledge what we don't know. So at least maybe someone in this room knows exactly what aging is and how it interacts with all different diseases, but I don't know. And if I don't know why this disease is so driven by aging, then how can I be sure that whatever model I create that includes aspects of aging, but not sort of like the full process, how can I be sure that that model actually has the right stuff? How can I be sure that I haven't created a model that sort of represents the disease if only it happened in 25-year-olds? And I think nobody sort of explicitly says, I'm going to go find a drug for osteoarthritis or for Alzheimer's that only works in 25-year-olds. But if we let our models basically represent that, if we let the disease biology of our models be sort of be in an overall young state, and therefore not having whatever age-related changes enables the disease to manifest, then we might end up doing that. So we have these horses that have developed osteoarthritis over a number of years, and then, as I mentioned at the beginning, we are injecting them with a range of different interventions. This is the part where, unfortunately, I have to be boring and sort of obfuscate what all of the different targets we're studying are for now. But just to give an example of some of the actual data we have, where from the full transcriptome, we have sort of custom bioinformatics that splits up every change that happens with the interventions into different disease-relevant pathways, and then we can evaluate for each of the interventions along each of the pathways, are we having a beneficial effect or not? And that also allows us to detect, potentially, this thing might have a good effect on the actual cartilage deposition, but might have negative effects on other aspects of the cells. We can use that to rationally guide potentially combination therapies where we hit more than one target. Nobody knows to what extent that is required for all these complex age-related diseases, but there is a relatively poor track record of hitting individual targets and modifying the disease. So going back to sort of beginning, how do you optimize the translation of the work that you're doing, whether we're doing basic mechanistic research or drug discovery? And I would say focus on what is the biology we want to study. Maybe you have a chondrocyte and you want to focus on making a chondrocyte healthier, but go the step of asking what are the main, like in normal biology, what are the main regulators of the cell's normal function? And we may not need to model all of these things. I'm not saying everyone should just like go do all your studies in all humans. But we should ask ourselves, are there signals that just always are driving what's going on here? In fibrotic disease, for example, you usually always have this immune signals that's driving fibroblast behavior. Is that something that we want to have present in our model system? and then look at everything we would like to model, and then try to see if there is a system that can actually include most of what you want, and if not, then just sort of keep that in mind. I'm also going to say, of course not, there's expenses and there's logistics, and not everyone has an in vivo screening platform, so I'm not saying everyone go do all your studies in some large animal spontaneous model, but I think there are, once we've asked the question of like what do we need to model, there are things that we can do. If you're studying biological mechanism, just some ideas here. I'm not sort of dictating what anyone should do, but FYI, JAX has a colony of H-Black6 mice. They're setting up a colony of the HET3 genetically diverse mice that are used in the intervention testing program. If you think that this genetic diversity might be an important thing to study because whatever you're doing is sort of like variant across populations, then you should tell JAX that you want a bunch of mice, because then they will set up a larger cohort that you can then later access and that everyone can access. Also, could you go and, if you're working at a university, is there a vet school, or is there a vet school nearby where you might be able to do some studies or access tissues from other species if, for whatever you're doing, mice are not the best model, right? Like, I think often we're like, well, it's not the best model, but what can we do? I think the dog aging project is a cool example here. Also, if you have a really cool strain of animal, then you should ask your funders for funding to just set up age colonies so that other people can also do aging studies in those systems. And then if you're doing drug discovery, again, can you, instead of like, you're going to have to do large animal work to get to the FDA if you're in the US, or the EMA, can you do that in a trial in pets with spontaneously occurring disease instead? Also think about the patient, most people 65 plus have multiple diseases. Should you really have multiple diseases present in your model? Should you be feeding them the drugs that the patients are going to be on? And then finally, should you be asking Felipe and Mahmoud to fund cohorts of non-mouse models as for aging, so that when we all, when everyone realizes the importance of aging biology and wants to do studies, including non-mouse models, we have actually aged animals available. Felipe, and you're in the audience, sorry, I'm not really calling you out, but I know that evolution is uniquely primed to do something like this. And then finally, penultimately actually, I'm going to take off my Gordian hat, no more Gordian, and talk about some nonprofit work done by Norn Group, where recently we put out this big spreadsheet that you can access here, that lists a bunch of facts about different age-related diseases, including what are the different animal models available, what are features of the patients, which might be a useful resource for starting to ask these questions. And then finally, I know I'm close to time, but not at time, Morten asked me to give an update on the Impetus Grants program, which probably many people are familiar with. It's a very rapid turnaround, low bureaucracy funding mechanism for nonprofit research into aging. So last year, we gave away about 100 grants, about 200,000 on average. Too early to say what the impact has been, but so far at least there's 2 conferences that happened, there's 7 preprints citing funding, and 2 publications, as well as clinical trials that are recruiting to test rapamycin and other aging interventions. It's been pretty cool to see publications already come out faster than most grant programs take to deposit the money into your bank account. So the updates are that there's now a new program director, Lara Nushna, who's here on the photo, not in Copenhagen. And she is going to take this program and keep it running in future years, which I'm very excited about because it seems like a useful thing for the field, but not something that I have time for doing. So she's fundraising, just announced yesterday, 14 more grants on specific topics, with funding in part by Evolution, as well as other donors. And then I want to highlight that Geoscience has a special issue going on right now for all the people who received impetus grants, where you can publish both positive and negative findings of high-risk projects. And so we're trying to make it so that if you want to do something risky, you're not discouraged by like, well, if I disprove my hypothesis, we can't publish it. And that's it.
Thank you very much, Martin. It was good to see you. We're running a little late here in Copenhagen, so I think we have to move on to the next speaker, but hopefully we'll see you next year.
All right, sounds good.
Thank you.
Bye.